Ever wondered how scientists manage to isolate and measure the amount of DNA in a cell? Welcome to the fascinating world of DNA assay and purification. The journey begins with the extraction of DNA from cells. Cells are broken down using a combination of physical and chemical methods. This process, known as cell lysis, disrupts the cellular membranes to release the DNA contained within. Enzymes like protease K are often used to degrade proteins that might interfere with the DNA. Next, the DNA needs to be separated from other cellular components. This is often achieved using a process called centrifugation. The cell solution is spun at high speeds, causing the heavier components to settle at the bottom. The DNA, being lighter, remains in the solution above, also known as the supernatant. Now that we have our DNA in the supernatant, it's time to purify it. For this, a salt solution is added. DNA loves to bind with salts, and this property is exploited in this step. The DNA salt complex is then drawn out using alcohol, typically ethanol or isopropanol. But how do we know how much DNA we have? This is where DNA assay comes in. There are several methods to perform a DNA assay, but the most common one is the spectrophotometric assay. This method involves measuring the absorbance of light by the DNA at a specific wavelength, usually 260 nanometers. The absorbance is directly proportional to the concentration of DNA. So by comparing the absorbance of the sample with a standard curve, the concentration of DNA can be determined. But remember, the purity of DNA is equally important. Impurities can interfere with downstream applications like PCR or sequencing. The purity of DNA can be checked by calculating the ratio of absorbance at 260 and 280 nanometers. A ratio close to 1.8 indicates pure DNA. In summary, the process of DNA assay and purification involves cell lysis to extract DNA, centrifugation to separate DNA, salt precipitation to purify DNA, and spectrophotometric assay to quantify and check the purity of DNA. It's a complex process, but each step is crucial in ensuring that we have high-quality pure DNA for our experiments. This intricate dance of science allows us to delve deeper into the mysteries of life, one DNA strand at a time. So, the next time you hear about a breakthrough in genetics, spare a thought for the intricate processes happening behind the scenes. It's not just about the discovery, but also about the meticulous steps taken to reach there.